right, thanks everyone. I wasn't sure quite how many people to expect at the last talk of the conference, but yeah, it looks like I don't get off easy with an empty room, so let's go ahead. So my talk is about a couple of different microbial ecosystems I've been studying in subglacial environments that we think are supported by lithogenic hydrogen. And I wanted to, I wasn't sure how short on time we'd be running by the end of the day, so I wanted to make sure and get my thanks out here right away. So Dr. Eric Boyd is my PhD advisor, and thanks to Mark, who serves on my graduate committee. Melody and Rebecca are members of their respective labs, who have each been helpful in the field work and the experimental work and the data analysis of these projects. So one of the frameworks I like to approach this project through is an astrobiological question of what makes a planet habitable. And we can kind of go through a few first-order indicators of whether a planet may or may not be habitable. So I've just taken these from the NASA Astrobiology Strategy document from 2015. And these are all, these are important considerations to make in terms of narrowing our search for habitable worlds. But in sort of through a more ecological lens, we can think about more second-order constraints on microbial communities. And the one I'd like to focus on today is that a microbial community needs a source of fixed carbon. There has to be, a base of the trophic pyramid. There has to be a population carrying out primary production. And studying these environments on Earth is sort of complicated by the fact that most primary production on our planet is carried out by photosynthesizers. So we can put up, this very simplified schematic equation of photosynthesis in which CO2 and water, through the energy supplied by photons, are transformed into both reduced organic matter, biomass, and oxygen, which is then, the oxygen is used to oxidize some of that photosynthate for energy. And so photosynthetic plants, algae, cyanobacteria are using energy from the sun, CO2 and water to get both reduced biomass and carry out their energy metabolism. If we look at something like an icy world habitat, where we have no current indications of ongoing photosynthesis, if we want to find microbial communities in this kind of an environment, we're going to be having to look for those supported by geochemical sources of energy. Something like methanogenesis, in which hydrogen and CO2 may react to form, again, reduced carbon that can be assimilated into biomass, and energy for metabolism. So, this metabolism has been described as a free lunch that microbes are paid to eat, as it produces both energy and organic matter. So again, just sort of the complication here is that it's difficult to find environments on Earth that are not supported on photosynthetically derived energy and organic matter. And so kind of a refinement of our search for analog environments on our own planet would be to look for those that favor communities supported by chemolithoautotrophic primary production. And so I've looked for these communities in subglacial environments. So beneath hundreds of meters of ice, you can reasonably assume that the influence of sunlight and photosynthesis have been excluded. And depending on the bedrock beneath these glaciers, there's not a whole lot of buried organic matter to support microbial communities either. And we know that some of these communities are supported by chemolithoautotrophs. So Eric and Mark have done work over several years demonstrating methanogenesis in some subglacial environments. We can see through C14 tracer experiments that these organisms are indeed fixing CO2. And we can show that comminution of subglacial rock is enough to produce the hydrogen needed as a reductant to support these communities. And so my thesis work is really focused on trying to identify whether differences in the underlying geology of a glacial system can influence the abundance and structure of the microbial communities in these systems. And so I'm getting at that through the lens of hydrogen production. Again, it's the key reductant for methanogenesis and acetogenesis in their fully autotrophic iterations. And In a subglacial environment or in water-rock interactions, there are a few different ways of generating hydrogen. I think we've heard some at this conference about how some of these mechanisms are still under investigation, but I just want to focus your attention on two key mechanisms. One is silica shearing by glacial comminution can generate silica radicals, which can split water and generate hydrogen radicals, which will react with each other to produce hydrogen gas. And there are also mechanisms by which ferrous iron can reduce water. And Stevens and McKinley did some work in the late 90s, early 2000s on low temperature water reduction by ferrous iron. And so regardless of our understanding of the exact mechanisms by which these processes happen, I think it's safe to build a hypothesis around the idea that in a system with higher silica content, higher ferrous iron content, with more of these potential mechanisms for hydrogen production, we may see indeed higher levels of hydrogen dissolved in the subglacial fluids and meltwaters. And we might expect that this environment would favor hydrogenotrophic primary production in the microbial communities associated with these environments. So in order to try and demonstrate this phenomenon, I'm looking at a pair of glaciers that differ in their bedrock geology. So one is Robertson Glacier here in the Canadian Rockies. And the local bedrock is primarily carbonates. There's some shales. There's a decent amount of iron in the sediments beneath Robertson, but low silica content. And so this will be my model system of an environment with low potential for hydrogen production. And I'll be comparing these to a glacier in Iceland. This is Kötlujökull, which is in southern Iceland and overlays, geologically speaking, relatively fresh basalt. So with a high silica content, high in ferrous iron, we might expect to see more hydrogen production after comminution of this basaltic bedrock than we would at Robertson. And so Everett Schock was kind enough to provide some hydrogen data from the Robertson system, and we can see there was about 40 to 50 nanomolar hydrogen in the outflow waters at the time his group measured it. Here's our mobile geochemistry lab in Iceland, and at Kötlujökull I was able to measure somewhere on the order of 400 to 500 nanomolar hydrogen in the outflow water, so roughly 1 order of magnitude increase. And this evidence was enough for me to continue with some cultivation work to look at whether these differences in available hydrogen between the two catchments actually translate to differential abilities of the community to utilize hydrogen as a reductant and to use hydrogen to fuel primary production. And so in my activity assays, I used a microcosm-based approach. I inoculated these with a sediment slurry and a medium designed to mimic the composition of the pore water, the outflow water at each of these glaciers. I made them anoxic and gave them a 20% CO2 atmosphere to favor autotrophy, added 500 ppm hydrogen, and then as near as I could get to a 1 millimolar concentration of various oxidants utilized in chemolitho-autotrophic metabolisms. And so the output from this experiment, as I'm sampling the headspace and using a gas chromatograph to measure the total amount of hydrogen in these microcosms over time. And I would expect to see in an abiotic control or a heat-killed control that the level of hydrogen stays relatively constant, decreasing only to the extent that I'm pulling it out during sampling. And that in an inoculated experimental microcosm, I should see maybe after a short lag period, start to see hydrogen oxidation. And so I have charts of this for each of the oxidants and each of the glaciers, and those just get really complicated to look at. So on the next slides, the charts you're going to see are, I've taken the maximum rate of hydrogen oxidation in terms of nanomoles oxidized over the number of days in the measurement interval. And I'll just jump to that. So first, here are the results for for Robertson, excuse me. So this is hydrogen oxidation. Across the x-axis, I have my control conditions, my experimental conditions with the oxidant arranged by relative redox potential. And then you can see on the y-axis the maximum rate of hydrogen oxidation observed in nanomoles per day, normalized to grams dry weight sediment. And so these are, I don't see really any significant patterns in differences among the various supplied oxidants in this pattern, or in this set of microcosms. But what I did see is a major difference between the Robertson sediments and the Kotliokel sediments. So this suggests to me that indeed at this basalt-based system, where we observe higher hydrogen production, we seem to see a community that is better adapted to take advantage of this geogenic energy source as a reductant fueling microbial metabolism. And the story turns out to be pretty similar if I look at CO2 fixation in these communities. So again, a microcosm-based approach, and to these microcosms, I've added a small spike of 14C, and again, the same set of oxidants, and I'll be looking in the next slide at, again, a maximum rate per day, maximum rate in terms of nanomoles fixed carbon per day. And these, again, are a subset of the rates that I observed over time as these experiments progressed. And so if we look at the hydrogen or the CO2 fixation results, we can see with Robertson, again, predominantly low rates of CO2 fixation, with the notable exception of microcosms amended with nitrate as an oxidant. And I interestingly see the same pattern with the Kotliokel sediments, Again, you can see if you look at on a pairwise basis at each of the oxidants that Kotliokel consistently supports higher rates of CO2 fixation. And again, we see this massive spike when nitrate is provided as the oxidant. And so, I'm still in the process of working through these results, and in particular, what it might mean that nitrate seems to support the highest rates of CO2 fixation. And one hypothesis may be that these systems are nitrogen limited. But in any case, I think I hope I've been able to convince you that we may be able to correlate the abundance of minerals high in silicates and ferrous iron with the potential for an environment to produce hydrogen. And we can use this potential for hydrogen production as a proxy for where we may find chemolitho-autotrophic microbial populations supporting broader microbial communities. So again, I've shown that in a basalt-hosted system, we see populations of microbes that are better able to utilize hydrogen as a reductant, and they're better able to couple that reductive or this hydrogen oxidizing metabolism with the ability to fix CO2 from the atmosphere, thus serving as sort of a base of a trophic pyramid for a microbial ecosystem. So in terms of future work on this project, we're working with the Skidmore lab on looking at the production rates of hydrogen from each of these sedimentary systems, and I believe that paper has been accepted. Is that right, Mark? It's on its way. You can talk to Mark about that project. And then I'm in the process of working through the last set of experiments I have going with these sediments is most probable number array. This is a monster set of inoculations in which I'm estimating the number of hydrogenotrophic and autotrophic cells in the sediments from each of these glaciers. And I'll be extracting DNA from these microcosms, attempting to correlate it with the metagenome data we have from each of these subglacial systems to learn more about the proportion of hydrogenotrophs in each of these microbial communities, and try to get some more information about the structure and assembly of such communities. So I'll leave you with a question. I'm looking for a name for my superhero alter ego. This is the suit you can see here. If you have any suggestions or other questions about the work, I'd be happy to hear them.
Thanks, Eric. We have time for some questions, if there are any for Eric. Sure, I think there's one back here, maybe. Oh, thought I saw a hand go up there. I have a quick question for you, Eric, regarding your results that you saw the highest rates of carbon dioxide fixation in the nitrate reducing populations. And do you think that's possible that it's just sort of a thermodynamic ladder thing, that nitrate's a really good oxidant, in terms relative to sulfate?
Well,
coupled to...
I mean, that is one hypothesis that sort of immediately jumps out. And I'm not sure how that relates to the rates we see for under oxic conditions in each of these sediment sets. One idea is that we may be seeing evidence that these are ecosystems in which the microbes are adapted to anaerobic, anoxic conditions, and perhaps nitrate is just serving as sort of releasing nitrogen limitation in these systems. It may be serving as primary oxidant for dissimilatory metabolism as well. But yeah, I'm not sure that I could say that it's just in relation to the redox ladder, judging by the fact that the oxygenic rates of hydrogenotrophy and carbon fixation seem to be so much lower.
Are you planning to redo this experiment and add maybe some nitrite or some ammonia?
That's always a possibility. One thing I would like to do in terms of rerunning this CO2 fixation experiment is to run some extra controls in which I don't provide any hydrogen, because I think that may help tease apart some of the patterns I'm seeing, especially with regard to early results of my most probable number experiments. But yeah, I would like to be able to at least confirm that this CO2 fixation is or is not related to hydrogenotrophy specifically.
Thank you.
Great, thank you, Eric. And thank you to all the speakers.